Let's talk about
little bit.
Interphase is a phase a typical cell spends most of its life. It is the daily living of the cell in which the cell obtains nutrients, grows, releases DNA, conducts other normal cell functions, and get ready for cell division.
This is prophase. In prophase, the nucleus dissolves and the chromatin in the nucleus condenses into chromosomes. Chromosomes with identical traits, known as homologous pairs, will undergo homologous recombination, where they swap parts of genes and DNA, so there's variation, so you don't look like your sister or brother.
Now the chromosomes begin to move towards the center of the cell to produce a straight line of chromosome. Now the homologous pairs align and the spindle fibers of the centrosomes emerge, and the chromosomes will line up and the spindle fibers grab onto the chromosomes using the kinetochores in the centrosomes.
The next step is anaphase 1. Imagine that A is for away or apart, because in this step, chromosomes are going to move apart, pulled to opposite poles by the spindle fibers. Telophase 1 is the last step of meiosis 1. You can imagine the T for 2 because it ends with 2 cells. However, we will end at more than 2 cells in meiosis 2. During this phase, spindle fibers disappear. After telophase 1, we are in interkinesis. Interkinesis is a period of rest that cells of some species enter during meiosis, between meiosis 1 and meiosis 2. There are no DNA replication occurs during this stage. Prophase 2 is the very first step in meiosis 2. We are not going to do interphase again. There is no more crossing over process in prophase 2.
During metaphase 2, chromosomes line up in the middle again and spindle fiber attached to each of those centromeres. This is the process of anaphase 2. The chromatids are getting pulled apart to opposite the sides of the cell and prepare to divide. Now the spindle fiber disappear and chromatids begin separating into individual gametes. The cells begin to divide.